Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we dealt with perfural derivatives and monosaccharides. In today's session, we will start with the disaccharides. In classification of disaccharides, first monosaccharides classification is given. Glucose, which is an aldose, galactose, which is fourth epimer of glucose, mannose, which is second epimer of glucose, and fructose, which is nothing but a ketohexose. Coming to the classification of disaccharides, glucose plus galactose give lactose, which is a reducing sugar. Glucose plus glucose is equal to maltose, which is also a reducing sugar, whereas glucose plus fructose is a sucrose, which is a non-reducing sugar. Coming to the next point, polysaccharides. These are polymerized products of many monosaccharide units. They may be homoglycans, which are composed of single kind of monosaccharides. Example, starch, glycogen, and cellulose. And heteroglycans, which are composed of two or more different monosaccharides. Example, hyaluronic acid and chondroitin sulfate. Coming to the structure of starch. It is a reserve carbohydrate of plant kingdom. Example, the sources are potatoes, tapioca, cereals, which are rice, wheat, and other fruit grains. Starch is composed of amylose and amylopectin. When treated with boiling water, 10 to 20% is solubilized. This is called as amylose. In this diagram, we can see the structure of amylose, which is nothing but glucosyl 1,4-glucose. Amylose is made up of glucose units, alpha-1,4 glycosidic linkages. They form an unbranched long chain with a molecular weight 4 lakh Dalton or more. The soluble part of water and forms paste-like gel. This is called as amylopectin. Amylopectin is also made up of glucose units but is highly branched and molecular weight being more than 1 million. The branching points are made up of alpha-1,6 linkage. In this diagram, you can see that this is the amylopectin structure, which is nothing but glucosyl alpha-1,6 glucose. Coming to the hydrolysis of starch. Starch is formed of blue colored complex with iodine. This color disappears on heating and reappears when cooled. This is a sensitive test for starch. Starch is non-reducing because free sugar groups are negligible in number. When starch is hydrolyzed, a mild acid, smaller and smaller fragments are produced. Thus, hydrolysis of a short time produces amylodextrin, which gives violet color with iodine and is a non-reducing sugar. Further hydrolysis produces erythrodextrin, which gives red color with iodine and mild reduction of pyrenic solution. Later, acrodextrins are produced, which give no color with iodine but are reducing. And further on, maltose is produced, which gives no color with iodine but are powerful reducing agents. Coming to the action of amylases on starch. Salivary amylase and pancreatic amylase are alpha amylases, which act on alpha-1,4 glycosidic bonds to split starch into smaller units, that is dextrins, and finally into alpha maltose. Beta amylases are of plant origin, almond, germinating seeds, etc., which split starch from beta maltose. They act on amylose to split maltose units consecutively. Thus, the enzyme starts action from one end. When beta amylase acts on amylopectin, maltose units are liberated from the ends of the branches of amylopectin until the action of enzyme is blocked at the 1,6 glycosidic linkage. The action of beta amylase stops at branching points, leaving a large molecule called limit dextrin or residual dextrin. Coming to the next polysaccharide, glycogen. It is a reserve carbohydrate in animals. It is stored in liver and muscles. About 5% of weight of liver is made up of glycogen. Excess carbohydrates are deposited as glycogen. Glycogen is composed of glucose units joined by alpha-1,4 links in the straight chain. It also has alpha-1,6 glycosidic linkages at the branching points. Molecular weight of glycogen is about 5 million. Innermost core of glycogen contains a primer protein called glycogenin. Glycogen is more branched and more compact than amylopectin. In this diagram, we can see that structure of glycogen, which contains multiple branching points, and at the innermost core it contains primer protein known as glycogenin. Coming to the third polysaccharide, cellulose. It is a supporting tissue for plants. Cellulose constitutes 99% of cotton, 50% of food, and is most abundant organic material in nature. It is made up of glucose units combined with beta-1,4 linkages. It has a straight line structure with no branching points. Each molecular weight is in order of 2 to 5 millions. Beta-1,4 bridges are hydrolyzed by the enzyme cellulose, but this enzyme is absent in animal and human digestive system, and hence cellulose cannot be absorbed in their system. Herbivorous animals have large cecum which harbor bacteria. These bacteria can hydrolyze cellulose, and the glucose produced is utilized by the animal. White ants, that is termites, also digest cellulose with help of intestinal bacteria. Cellulose has a variety of commercial applications, as it is the starting material to produce fibers, celluloids, nitrocellulose, and plastics. Coming to the fourth polysaccharide, inulin. It is a long-chain homoglycan composed of D-fructose units with repeating beta-1,2 linkages. It is the reserve carbohydrate present in various pulps and tubers such as chicory, dahlia, dandelion, onion, garlic, etc. It is clinically used to find renal clearance value and glomerular filtration rate. Inulin and insulin are different. Inulin is a polysaccharide that is carbohydrate made up of fructose units. It is used for renal function studies, whereas insulin is a polypeptide that is a protein or hormone with wide-ranging actions on carbohydrate and lipid metabolism. Coming to the fifth polysaccharide, dextrans. These are highly branching homopolymers of glucose units with 1,6, 1,4, and 1,3 linkages. They are produced by microorganisms. They have molecular weight 1 million or to 4 millions. They are used for infusion, intravenous infusion as plasma volume expander for treatment of hypovolemic shock. One should not get confused between dextrose, dextrin and dextran since they are different. D-glucose is otherwise called dextrose, a term which is often used in the bedside medicine. Example, dextrose strip. Dextrin is a partially digested product of starch, whereas dextran is a high molecular weight carbohydrate synthesized by bacteria. Coming to the sixth polysaccharide, chitin. It is present in exoskeletons of crustaceae and insects. It is composed of N-acetyl glucosamine with a beta-1,4 glycosidic linkage. Coming to the heteroglycans, they are polysaccharide containing more than one type of sugar residues. Agar. It is prepared from seaweeds. It contains galactose, glucose, and other sugars. It is dissolved in water at 100 degrees centigrade, which upon cooling sets into a gel. Agar cannot be digested by bacteria and hence used widely as supporting agent and to culture bacterial colonies. Agar is used as a supporting medium for immunodeficient and immunoelectrophoresis. Agar is made up of galactose combined with 3,6-anhydrogalactose units. It is a matrix for electrophoresis. Coming to mucopolysaccharides. Mucopolysaccharides or glycosaminoglycans are heteropolysaccharides containing uronic acid and amino sugars. Acylated amino group, alpha sulfate and carboxyl groups are also generally present. Because of the presence of these charged groups, they attract water molecules and so they produce various viscous solutions. Mucopolysaccharides in combination with proteins form mucoproteins. Examples of mucopolysaccharides are hyaluronic acid, heparin, chondroitin sulfate, dermatan sulfate, and keratan sulfate. Coming to 1. Hyaluronic acid. It is present in connective tissues, tendons, synovial fluid, and vitreous tumor. It serves as a lubricant in joint cavities. It is composed of repeating units of N-acetyl glucosamine, beta-1 glucuronic acid, and beta-1,3 N-acetyl glucosamine, and so on. Coming to the second one, heparin. It is an anticoagulant widely used when taking blood in vitro for clinical studies. It is also used in vivo in suspected thromboembolic conditions to prevent intravascular coagulation. It activates antithrombin 3, which in turn inactivates thrombin, factor 10 and factor 9. Heparin is present in liver, lungs, spleen and monocytes. Commercial preparation of heparin is from animal lung tissues. It contains repeating units of sulfated glucosamine and alpha-1,4-L hyaluronic acid and so on. In this diagram we can see the structure of heparin, which is nothing but sulfated glucosamine alpha-1,4 hyaluronic acid. Iodose is the fifth epimer of glucose. Hyaluronic acid is the oxidized form of iodose. Sulfated heparin or heparan sulfate is also present in tissues. Here ends our session. Kindly like, share and subscribe the channel. Thank you.